There's one more bacterial structure that we have not yet discussed, and that is a structure known as an endospore. So this is what an endospore actually looks like. It is a survival structure that is formed by certain bacteria. So not all bacteria can form endospores. Let's take a look at how this process happens. So we're going to look at this in cartoon form. Imagine that you are a single bacterial cell and you find yourself in a stressful environment. So for a bacterial cell, stress isn't a big exam. Stress more comes in the form of maybe you find yourself in a really dry environment where no water is available. Maybe you're in an environment where there's no nutrient source, no energy source. And when a bacterium finds itself in an environment that is lacking in one of the requirements for life, and it knows, quote unquote, that it's about to kick the bucket, what will happen is it will form this structure called an endospore inside itself. So it basically makes a copy of itself and wraps it up in armor so that when this bacterial cell dies and it spews its guts out into the environment, what remains in the environment is this structure called an endospore, or sometimes you'll hear people just call it a spore. And this structure is incredibly resilient. So it's kind of a way for the bacteria to hunker down until the conditions in the environment improve. And when the conditions in the environment improve, it will shed these layers and kind of reanimate and grow back up into the happily metabolically active bacterial cell. So let's jot a few things down. Endospores are structures that are formed only by some soil or gut-dwelling bacteria. And they do this in response to stress. So not all bacteria can form endospores. More specifically, it's organisms in the phylum Firmicutes, in the genus Clostridium and Bacillus. And so spores are often found in the environment. And this is one of the reasons why, or this is the primary reason why, you don't give a baby honey, right? Because sometimes some of these spores can be found in honey because there are bacteria in the environment that are capable of forming spores. It's also important to note that Clostridium represents a really medically relevant genus. So some of the nosocomial or hospital-acquired infections occur with the genus Clostridium, specifically Clostridium difficile, or C. diff, which you guys have probably heard about. So if you've ever worked in a hospital setting, you'll be trained on how to deal with endospores because Clostridium is known to be able to form those. What an endospore is a metabolically inactive bacterial cell. So that initial bacterial cell makes a genetic copy of itself that is metabolically inactive. And then it wraps that genetic information with protective proteins. So those different layers that we showed initially, those are made of protein. And that is like the armor around the genome, right? That's the armor that surrounds the genetic information and protects it. Spores are incredibly resilient. They can withstand temperatures up to 150 degrees or 302 degrees Fahrenheit. They can withstand drying conditions. In fact, spores often form when there's a lack of water in the environment. And that kind of facilitates the shutdown of metabolism. Spores can also withstand UV radiation. And these things are really significant when you're trying to sterilize surfaces, particularly in a hospital room where you might have an immunocompromised patient. There's some terms that we should kind of familiarize ourselves with that are associated with endospores. The first is sporulation. So sporulation describes the process by which a bacterial cell makes an endospore. Germination is when that endospore kind of wakes up to become metabolically active again. So when an endospore germinates, that means that it's kind of reanimating and becoming metabolically active again. And finally, the last term you should know is vegetative cell. So a vegetative cell describes the metabolically active cell. So the vegetative cell is the cell that, when under stress, may be capable of forming an endospore. One distinction I really think it's important to make is that sporulation is not a form of reproduction. Sometimes you might see people refer to the vegetative cell as the mother cell, and that kind of implies that a mother cell is reproducing itself, but that is not the case. This is purely a survival strategy. So let's take a closer look at an endospore and kind of identify some of the important components. So now we're depicting our bacterial cell in kind of an oval shape here. And inside you can see the chromosomal DNA. In order to perpetuate itself, it really has to protect this chromosomal DNA. And it does that with a couple of important molecules. So the first molecule we're going to learn about is called dipeclinic acid. And dipeclinic acid is complexed together with calcium. And it's the role of dipeclinic acid and calcium to protect DNA. And that makes sense. You'll remember from cell biology that the sugar phosphate backbone of DNA is negatively charged, and we know that calcium is positively charged. So that allows the dipeclinic calcium complex to bind to the DNA and protect it from any kind of enzymatic degradation. In addition, the dipeclinic acid and calcium complex also helps to dehydrate the cell. And that's really important because in order for a cell to shut down metabolism, you have to get all the water out of the cell. The second molecule that's really important in protecting the DNA is called a small acid soluble protein. And you can call them SASPs for short. So SASPs also help protect the DNA, but in addition to protecting the DNA, they also act as a nutrient source. So I like to think of SASPs as an endospore's packed lunch, because eventually, when that endospore germinates and becomes a vegetative cell or a metabolically active cell, it's going to need some kind of energy source in order to make that transition. So this is what an endospore looks like on the inside in cartoon form. And you may be wondering what it is that causes this metabolically dormant cell to suddenly wake up. What are the germination triggers? So to understand that, let's go ahead and put our layers of protein on the outside. So we have lots and lots of layers of protein that are surrounding this DNA and protecting it from the environment. And it's these layers of protein that allow this endospore to withstand such high temperatures, UV radiation, and other things, because there's lots and lots and lots of layers. Embedded in these layers are receptors. So these receptors, represented here in green, are capable of detecting germination triggers, or molecules that tell that cell to become metabolically active again. So let's go ahead and label these guys. These are the receptors for germination triggers. And the reason why we have germination triggers in quotes here is because there's lots of different molecules that can cause a spore to germinate and become metabolically active. So essentially molecules that do that are those that indicate that good times are here again, right? Certain nutrient molecules or water molecules, amino acids, sugars, might bind to their cognate receptor and tell that spore to become metabolically active again, to come on out and reanimate into a vegetative cell. So when these germination triggers are bound, that triggers the breakdown of these outer protein layers. So this entire thing, this entire structure here is a spore. And when a vegetative cell finds itself under stress, it'll make one of these. And so when that cell then succumbs to the lack of nutrients or the lack of water, it's got a copy of itself encased in this armor, this protective shell, and with molecules protecting its DNA, and it'll just kind of hunker down. And spores can hunker down for a really long time. I'll show you that in just a moment. I think it's really important too that, we don't just see this in cartoon form. So let's take a look at an electron micrograph of an endospore. And you can see a lot of the structures that I was telling you about. So all these layers here, all these layers around this kind of inner oval are the protein layers. And inside here is the DNA. It's the genome of that bacterial cell. And you can't really see the dipicolinic acid calcium complexes or the small acid soluble proteins, but they're in there protecting the DNA. When you guys work in the lab, you may be doing an endospore stain. And there's different kinds of endospore stains. But looking under a compound microscope, this is one way that these endospore stains may look. So some stains will actually color the vegetative cell, kind of a purple color. And it's the absence of that purple color, the absence of stain, that indicates the presence of endospores. You can see that some of these endospores are still inside the vegetative cell, and other endospores have actually been released into the environment. And the other thing I'll have you notice is that this is a bacillus-shaped bacterium. So presumably it's some species from the Bacillus genus. The stain that we do here at Shoreline looks a little bit more like this. So the vegetative cell will stain pink, and the spore will stain green. So this is a type of dye called malachite green, and you'll actually steam your slide to have this stain penetrate the spore. So you can see that most of these bacteria are still holding on to their spore, but a couple of them have been released, like this guy here, and this guy over here. See these guys here. So this kind of tells you something about the age of your bacterial culture, right? So the older your bacterial culture is, the more likely that it's going to run out of nutrients, and the more likely that those bacteria are going to start forming these survival structures. Knowing that bacterial spores are so incredibly resilient in really adverse conditions, you might be wondering how long spores can remain viable. And this question was best addressed back in 1995 when there was a group of researchers that had found some extinct bees encased in amber from a particular type of tree in the Dominican Republic. So they were able to crack open this dried amber and then dissect out the gut of these bees. And in their gut, they found endospores from a species called Bacillus sphaericus. And when they recovered these endospores, the group claimed that they were able to germinate these spores back into vegetative cells. So that research was kind of controversial because there were some groups that thought that there's no way that 25 to 40 million old endospores, DNA in the endospores could be still functional. And they claimed that it was actually due to contamination. So the jury's kind of out on this one, but it's still pretty fascinating nevertheless. So here's what that looked like. Here's a bee that's encased in amber, and they were actually able to get into the gut of that bee and pull out those 25 to 40 million year old endospores that were formed by bacteria.